Optimizing Cell Number for MTT Assay by Giselle and Jeff. Let's begin with sterile technique. Proper aseptic technique is imperative to your experiment. This includes, but is not limited to, washing hands before and after, wearing your lab coat, wearing gloves, spraying down your gloves, utilizing the hood properly, spraying down equipment and hood surfaces, and using the right tools for the task. Prevention of contamination can keep your experiment on schedule. Remember, contamination can set your experiment back a week, as you may have to clean out the incubator and wait for more cells to grow. Wearing a clean lab coat, one that is not used in other labs, is important. Leave your lab coat in the storage cabinets if you're concerned about cross-contamination. Remember, you are both the biggest potential cause of contamination as well as the first line of defense. After you have put on your safety equipment, remove your media from the fridge and place the media in the 37 degrees centigrade water bath. The media will need about 20 to 30 minutes to get to the proper temperature. This is important because cells are incubated at 37 degrees. By allowing the media to get to the proper temperature, you help make sure that the cells are not temperature shocked once media is added. Spray 70% ethanol solution on your hands before opening the incubator. Carefully remove your flask. Check your media for clarity. You should be able to see through the media despite having a lot of cells. Place your flask under the microscope. This is the primary way to check for contamination. Next, we talk about using the hood. Before using the hood, make sure that the UV light is on. Gather the materials that you need to perform your experiment, such as pipettes and tips. Lift the sash and make sure it does not go above the sash level. Wait for the fan to start blowing. Listen for the fan. Once the fan is running, spray down the hood with the 70% ethanol solution and wipe it down. Remember to spray your hands before placing them into the hood. Remember, never leave any cells, media, compounds, or reagents under the UV light. Spray down all items before placing into the hood. Obtain your cells from the incubator when you are ready to begin the experiment. Remember, placing cells in the hood should be the very last step before you sit down. When placing your hands back into the hood, remember to spray them with a 70% ethanol solution. Before we take a sample, check the hood for a waste bottle containing bleach, a beaker for holding tips after use, and a beaker containing autoclave Eppendorf tubes. Now, obtain an autoclaved Eppendorf tube. Keep the lid closed until use. Open the serological pipette tip at the filtered end. Insert into the pipette. If necessary, turn the pipette so you can see the numbers, but do not remove the sleeve. Now, remove the cap from the flask and place face down. You can pick up the pipette, and now you can remove the sleeve. Remember to save the sleeve for later, as it will be used for disposal. Place the tip into the flask and pipette the entire volume of the flask. Wash the back wall of the flask with the culture. Repeat this 2 more times for a total of 3 times. Note, you should only ever use the serological pipette inside of the flask. Do not use any of the blue pipettes. Try to avoid bubble formation when pipetting. Now obtain a half a milliliter sample and place it in the autoclaved Eppendorf. When you are done using the pipette tip, place it back into its sleeve for disposal. Remember to also put your flask back into the incubator when not in use. Before counting, reset the cell counter. Make sure that your pipette is set to 15 microliters. Obtain an accurate sample by placing the pipette tip in the center of the cell culture. Before loading your sample, make sure that your hemocytometer and glass cover slip are clean. Then place the sample into the triangular groove. Dispose of your pipette tip. Now place the cover slip carefully on top. Watch as the sample fills the grid. Then place your hemocytometer on the microscope for counting. Begin with the lowest magnification to find the grid, then adjust as necessary to view the 16-square quadrant. Here's an example of what your plate would look like. Use the first two columns. This is performed as a duplicate to ensure reliability. Here's a recreation of the table found in your handout. Now let's take one of those columns to calculate. Convert the number of cells into a percentage. Then convert the percentage into microliters of cells. From this, you can now calculate the volume of media you'll need. Ultimately, every well should have a total volume of 100 microliters. Now add these volumes together to obtain the total volume per column. Times 2 columns is 455 microliters of cells that you need to obtain. Now gather your media, empty tube for cells, pipettes, and pipette tips. Obtain your cells last. Prepare your pipette as before. We will use it to obtain a sample from the cells. Open the flask and place the cap face down. Resuspend your cells at least once before taking the sample. Now obtain the volume of cells you had previously calculated. Taking a bit more is okay. Recap your flask before continuing, and pipette your sample into the empty tube. Make sure the cap on your flask is tightened and return your flask to the incubator before continuing. Obtain your plate so you can begin plating as per your schematic. Begin by first pipetting the media into the plate. Change pipette tips as necessary to prevent contamination. Using a fresh pipette tip, pipette the cells as outlined by your calculations. When you are done, place the plate into the incubator and allow to incubate for 48 hours before beginning MTT. Obtain your light-sensitive MTT reagent housed in its black tube. Pipette the correct amount according to your handout. When you are finished dispensing your MTT reagent, place the plate back into the incubator to allow to incubate for 3 hours. Your plate should now have a yellow color due to the reagent. After incubation, you can add detergent on the benchtop. Add 100 microliters to each well. Once again, you can confirm the addition of the detergent by the change in color. When you're finished dispensing the detergent, wrap the plate with aluminum foil and keep it in room temperature for about 2 hours. After incubation, place the plate into the microplate reader to obtain your readings. Upon completion, place your plate into the biohazard bin. Now to analyze your data. Tabulate your absorbance values on an Excel sheet. Here are sample absorbance readings by the spectrophotometer. The number of cells per well should also be calculated using your cells per milliliter count and the appropriate percentages. Calculate the average absorbance. Standard deviation is calculated using the two absorbance values. Now let's graph the data. Create a bar graph showing average absorbance versus number of cells. Use standard deviation to create the appropriate error bars. Look for absorbance values within the range of 0.5 to 0.8. This is because of the way cells grow. Looking at the graph of the growth curve, we can see that we want to be after the lag phase, but before the stationary phase, right in the log or exponential phase of growth. If necessary, take the average of two cell densities as you would have to do for this sample data.